bed goes from here for quite a while. That's another one of these very conspicuous anemones. We've imaged a couple on these dives already, and that's going to be in the genus EX1603.
Got some really neat landscape imagery that we're getting right now. As you can tell, there is a couple of identifiable layers that are right in here. So we've been going over maybe 2, maybe even 3 individual lava flow layers. They're dipping downhill, down to the left. Just some spectacular basic stratigraphy for a good image on. And you can kind of see how sharp this actual ridge is. So we're literally riding the ridge crest, and it's not more than a meter or 2 across before you're going downhill one way or the other.
5, please.
Copy that.
Yeah, and it also gives you a really nice appreciation for the narrowness of this feature. So this ridge is a very, very narrow feature. It slopes off on both sides. And there's several habitats like this here in the northwestern Hawaiian Islands. And one of the unique things is that we have several of these rift-zone ridges that extend over a very long time, and then presumably providing continuous habitat for many organisms. And yeah, one of the things we're trying to understand is how do these dense communities form? Where do they form? What species are they composed out of? And then hopefully know and find out enough about these things so that we can model them and then try to predict where we might find them. So yeah, again, a lot of primnoid corals, so those sea fans. There's a stock crinoid. There's another Chrysogorgia.
Copy that. Thank you, Ridge.
Very beautiful imagery.
Now let's actually tell them to add 15 to the end of that.
So just looking ahead, we have
Bridge, this is
just under an hour left on this dive.
We'd like to add another 15 on to the end of that move.
We've been on the bottom a little over 4 hours now, and which has truly been a remarkable dive.
Lost it.
And Scott also made another interesting comment. So yeah, one distinctive feature about this dive in comparison to many others is that, well, one, we've seen a lot of primnoid corals and a lot of small corals, and in most other places where we've targeted these kinds of depth ranges, we've seen mostly bamboo corals. On this dive, we've seen very few bamboo corals. So we see several black corals here. There's Stauropathes just to the top, to the left and right, so several colonies.
Come on in video.
Say again?
It's come on in.
Roger, coming in.
Yeah. Sorry, I'll speak up.
Oh, excellent. Yeah, that sea star is going to be again of interest. Good eye. And I believe that is once again going to be Henricia.
It's a full zoom.
Thank you, Bridge.
On the substrate itself.
Come out. Is there anything else you want to see or watch, Lee?
No, that's okay. Thank you. But yeah.
Full wide. Minus skids.
One interesting thing also about today's dive is that it's been a fairly low density, low diversity of sponges. So in a lot of these dives we've seen quite a remarkable diversity. So the vast majority of the sponges we've seen on this dive are these farreate sponges in the genus Spidoscopulia. And beside that, we've seen just a few other exactinellids, but not really a high diversity. And then we've seen a lot of this dead sponge material.
That's about all you're going to get
That's it.
from my tether.
Come up, look at the other side.
Yeah, if we have a second here, maybe just a quick zoom on that primnoid that's right of center.
Yeah, copy.
Dead ahead.
Come on in video.
Coming in.
So that's going to be another primnoid octocoral.
And come more if you want.
Yeah, go ahead.
And looks like it's in the genus Narella. They do have the downward facing polyps, which are characteristic for this group, this genus.
The move will be completed here shortly. Do we want to line up another one or just kind of wait?
Attention all hands, the operations meeting will be held in the forward lounge.
And
5 minutes at 14:30. Operation
it's
meeting in the forward lounge in 5 minutes.
so my best guess here, based on this image, is a Narella macrocalyx.
I don't have a clear
There he
direction
is.
forward, Nav.
I'll come up after this and
Watch that.
take a look.
Is there any rack to that further one?
And just to provide those viewers that might have joined us later in the dive, yeah, that's wonderful. Thank you. So
Come on out.
we spent about 4 hours now in the bottom. Transit about 600 meters on this rift zone ridge. Collected 2 geological samples and 2 biological samples, which are going to be very valuable to analyze in the laboratory. The coral samples in particular are like nothing we've seen before in this region. So at the very least, they're going to provide new records for this region. Not that much in terms of fish that we imaged, but we were able to capture a couple of synapobranchid eels as well as a macruit.
Thank you, Bridge.
And have been basically
Yeah, I think still north.
meandering around this crest,
You
imaging.
I do.
You want to
It's
do
kind of
30
been
meter?
continuous moderate densities of animals with patches in between where you
30
see
sounds
really
good to me.
high densities.
Copy that.
Yeah, we can do 30. 355.
Yeah, sounds good.
Bridge, this is Nav.
Let's come in just a little bit, lose the
I'd
skids.
like to request another move at 30 meters. That's 30 meters at 355 at 0.2 knots.
There's another fish.
Another rat tail.
And so that's
Come
going to
on
be
in, video.
another
Coming in.
rat tail or
Good count.
and
Little
or
bat.
mercurid. And I'm sure Bruce is watching very attentively, as is probably
Lost the
Astrid.
thruster. I'm standing by to do it. Okay.
Thanks, Bobby.
And it looks like that's same Kumba to me, but I'll wait and see what Bruce and Astrid have to say. Astrid thinks it is. Yeah, that rounded snout.
Are you chasing him?
Yeah.
Again, these
I'm gonna let him
mercurids,
go off.
Copy that. Thank you.
also known as rat tails
That would explain how he was
or
swimming
grenadiers,
away, yet
are some
getting
of the most
closer.
Hey, we
abundant
can come wide.
Alright, go wide.
fishes in the deep sea.
Camera
Full
is
wide.
090.
Copy,
They
090.
include some species that are also commercially harvested.
Rid of the skids here. Skid free.
So yeah, thank you, Astrid and Bruce, for providing your expertise. And Andrea as well. So for those folks who might be watching this in one of the general displays, we have 2 onboard scientists here on the ship, but then we actually have a whole fleet of shorebait scientists that are also watching the video in real time, and they're then providing help with identifying things and providing guidance on where to go, what to collect. And so yeah, it's all a remarkable product of this telepresence technology. So it does not only provide a way for folks just to chime in and watch this via the internet, it also provides for a very valuable collaboration tool, being able to pool resources that would otherwise not be available.
There's a big one that fell over, huh? A couple of them.
And yeah, Scott just observed that the fleet of scientists is on shore. Yeah, we have been lucky today, so we had a couple of rough weather days, both yesterday and the day before, facing 20-foot swells plus in the area that we're trying to target. So luckily today we got a break in the weather and able to dive on this feature. That's a very remarkable feature, and hope you all were able to enjoy it as much as I have. That's another one of this massive dead sponges that's dead. And yeah, that's a beautiful view of just how narrow this ridge is at the top. Out there in the distance is one of the most bizarre looking sponges. These are in the genus Bolosoma. They kind of look like ETs when you look at them up close. The top, the head of the sponge actually has several holes than you typically have. Shrimps and ophiuroids and a variety of other invertebrates living inside the head. And that's something that's very common, that you see a lot of other invertebrates living inside both corals and sponges, and And that's really why we're trying to target a lot of these areas where we think we're going to find high density communities. Because once you see these massive habitat forming species, you typically find other all kinds of other creatures living in association with them as well.
Do a quick zoom here.
Roger, quick zoom.
It's barnacles, I assume. Yeah.
Yep.
Yeah, so those are a couple barnacles, and for most of the barnacles we don't have good IDs.
Alright, when you're happy, watch lead and video.
30 meters due north.
Copy that.
We're just
Coming out.
So that's so far
I'd like to request a move of 30 meters.
the
due
best
north
I can do there is
at
take that
0.2
to
knots.
family, and that's going to be in the family, the Balanoidae. And barnacles are a
Good
problematic group. And yeah, not much has been done with the species here in Hawaii. And interesting, last year we found during our dives many places where all you could see were barnacle plates, so massive grounds all covered by these barnacle plates, suggesting that a lot of places they are quite abundant, and so we saw a lot of these barnacle plates in many of the places that we dove on, and all of us were asking ourselves, where are the barnacles? And it took us many dives later in the expedition until we actually found these dense aggregations of the barnacles.
Snap zoom here.
Snap zooming.
I think we've probably seen this guy. Oh, he's all closed up.
Yeah, so that's an anemone, and it's actually yeah, will be hard to get an idea with it closed up.
Yeah, if you got any more video.
I think that's about it.
Alright. You can come on out.
Just hold on a sec.
I'll hold it.
Just keep going to your right.
Oh, okay. Copy.
That's all right.
Couldn't get it out.
See if we can do something with that. Thank you. Oh, that's interesting.
So I just updated our position. We're about 650 meters
That's pretty well. That's right.
north of where we started to dive.
It kind of seems like this this crack that's running down the axis of the ridge might be separating flows that went down either side, actually. Very, very neat feature.
There's another one of these large brown anemones. That's going to be in the genus EX1603, one of the more conspicuous components down here in the deep waters. a lot of primnoid corals, so that's all those white small colonies, and a lot of dead sponge material, and a lot of these cracks we've been able to look into. We see a lot of this dead sponge material. Current depth is 2,338 meters, so we're actually not that much shallower there where we started to dive. This portion of the ridge is pretty much parallel to the surface. We haven't got much height.
Yeah.
So we started to dive only about 30 meters deeper than what we are right now. But we have covered some ground. Can we take a quick look at that black object in the bottom?
Bottom center screen.
Yep.
Right now. Yep. The discoloration.
Yeah.
Yep. All right, video.
Coming in.
Ah.
I'm just holding.
I gotta back up. I came over.
There you go. Coming in again. Holding.
I think you're all the way down on your camera.
Yeah, I am. I'm just getting pushed around here. Right.
What am I looking at? So, yeah, Tina has noted the C-pen in the back, and yeah, that's Protoptilum. But
I think that's geology.
Maybe that was there was a base of something attached there, and it
yeah, it came loose.
Came loose.
This some kind of discolored rock. Yeah, it just caught my eye, but I have no idea what I'm looking at. Yeah, and Scott agrees with you. Probably something was attached to this at some point. Now there is a C-pen just right of us, and we might be able to just quick snap soon. If you just pan a little right. Yeah, right there. And
this guy?
Lost my toe.
Looking down.
All right.
80 degrees.
Let's do a quick snap zoom there.
And that's going to be in the genome's Antoptilum.
All right, come on in.
Holding. Which we're going for, on the right?
The left. C-pen.
Yeah, so that's the first record we have of this guy in this depth. It's a rock pen, as they're commonly known, genus Antoptilum.
Move is complete.
Very interesting animals that
090.
look like these wind socks that you sometimes see at car dealerships.
All right, if you're happy, watch lead.
Yeah, thank you.
All right, come on up, video.
Yeah, that's a beautiful view of how narrow this feature is. Yeah, and Tina just made a comment that it didn't look as crooked as the Anthoptylum as other specimens, but we have imaged things just like that.
So I don't know if I can be sure, but that doesn't actually look like sediment. That looks more like a catchment of dead sponge tissue.
Yeah, so maybe we can take a quick look. So these are kind of these, we saw stuff like this.
Watch
and
my port swing arm here, Bobby.
Watch it. As long as you don't turn starboard, you'll be fine.
Alright, come on in video.
Coming in.
Yeah, little skeletal pieces of.
yeah, all kinds of debris.
I wouldn't say that is actually.
No, it definitely was part of an animal at some point, but.
Gonna lose you just for a second.
But that's different from those barnacle plates that I was mentioning earlier, where we saw piles just like this in many dives, but they were clearly plates. These look a bit more diversity of shapes. And Tina thinks they are barnacle pieces.
Zoom
Actually,
out for scale if you want.
thank you, that's great.
Yeah, come on out.
Slow out. Full wide.
So just to update everyone, so we are doing a normal recovery, so we'll probably be leaving the bottom in around 30 minutes or so. Currently going through a personnel change in our piloting team.
Sirius is 090.
So just hang
Way
tight.
forward to north.
So yeah, our current depth is 2,336 meters. We've covered quite a bit of ground on today's dive, collected 2 geological samples, 2 biological samples, so all the collections,
090.
all the collections we hope to do on this dive.
Due north.
And so we're just trying to cover some more ground on what's remaining in this last 30 minutes of this dive.
Do you want to pull ahead a little bit more?
Well, you look at that thing in the water column for a little bit.
Sounds good.
Josh will be right back.
Perfect. Yes, Fletcher, we have about 25 minutes left before we start to get ready to come up bottom.
Roger that, Nav. Can we take a look at that object that's in the water column just above us?
Coming up to it now.
Yeah. So,
Evasive. Maneuvering. Just like that. He's
yeah.
Don't see it.
That's. And Bruce thinks it could be and Bess has said is.
I don't mean I'm gonna chase it. I just showing you. I think it was over there.
So that is
That
an.
little thing? Nah, that wasn't it.
That's an Ophidid.
I think it was.
And I would definitely go to Ophidid.
That floating object came from below me.
So I mentioned that we have about 25 minutes remaining on this dive. Looking ahead, so there's basically several storm systems that are north of Midway that are really preventing us to go much further north. So we're going to try to dive again somewhere in this vicinity. There is a site that we had tentatively identified just north of us between Pioneer Bank and Lysianski. There's a couple of targets that we might try to get just south of us. So yeah, if the weather cooperates, we will get a dive in tomorrow and try to get this in this kind of latitude.
30 meters, 345. Copy that.
And we'll have a call to discuss that a little after the dive.
Just go and push in and lose the skids here.
Sounds good to me, video.
I'm in. Skid free.
Thanks. That's about as much as I've got. Cable, so we'll just spin around here and look around. Vehicle feels a little heavy. I haven't pushed down at all. Just been floating around.
So yeah, we're coming up on another little rock, and this is something that we've seen time and time again throughout this dive and previous dives, is that you have these little topographic highs, these elevated either boulders or pinnacles, and as soon as you see those elevated structures, they're a lot of times that provides prime habitat for corals, and you see them completely overgrown. We have several black corals there in the back. We have several primnoid corals in the front. That elongate coral to the left is a chrysogorgid coral. Those very interesting looking sponges there in the background that kind of look like lollipops. Those are going to be in the genus Bolosoma. And and then that chrysogorgid coral, that bottle brush shaped one to the left, you can see a squat lobster on it. So those are, it's likely going to be in the family Chirostylidae. And so that's a very common association, that you see squat lobsters perching themselves on top of corals and sponges. And so yeah, a lot of these invertebrates use these corals and sponges as habitat. And a lot of them use them kind of as platforms. If you were to measure current flow right at the bottom, right at the contact between those rocks and then the bottom, there would be 0. And then as you go up, you would start seeing an exponential increase in current flow with elevation to the substrate. And so it's beneficial for a lot of these organisms to get higher up into the water column, so they basically have more flow over the tissues that they can use to capture food or ventilate themselves. Yeah, if we have a second, let's maybe take a look at the larger fan that's right in front of us. It'll be hard to see from this angle, but we'll take what we can get.
I can get you a good angle. Oh, I have a rock behind me. Did not see that.
Yeah, Hefe brought you down on the right side of that overhang.
Alright, come on in, video.
Coming in, aye.
So that's Chrysogorgia, and it looks like Chrysogorgia chrysaris. So this planar Chrysogorgia, and we had not recorded that on today's dive.
Whoop, coming out. Did we lose our toe?
Not sure what happened there. We're good. We're good. Come on in.
Coming in.
Yeah, let me know when you're happy. Watch lead.
Yeah, no, that's good. Perfect.
Okay. Let's just get a nice shot of it, and then we'll move on.
So this group is characterized, so this family, the Chrysogorgia, by these very unique polyps. You see them kind of elongate, being set apart from the main stem. And you can almost pick that out even when you look at the colonies from a distance, is that it gives it a very unique appearance. Now, on the last dive, we collected a Chrysogorgia that
Coming out.
is very, very unusual.
Roger.
And on today's dive, again, we collected one of these, and that's, again, unusual. So it appears to be a very species-rich group here in the deep sea, and we've only described a handful of species. And so there's a lot of these forms that don't match any of the formerly described species, so a lot of work to be done.
Still heading north, Bobby?
Still heading north. Last move was 345.
And actually, so that coral that's right on top of the ledge, if we have a quick snap zoom.
The one with the long stem?
Yep.
Yep. All right, come on in, video.
Lost a thruster. Standing by.
Yeah, go ahead, reset.
So that is a different group of, so that's Chrysogorgia flavescens.
Give it back.
So it's one of these few species of octocorals that has that kind of plume up top with a.
That's good.
With the stalk.
Did you have no control?
I had no control.
And nice back drift. We have a.
Thanks.
Yeah, it's exactly what I wanted.
I had no control during that entire drift.
Yeah.
That was strange.
Yeah.
We got it.
Maybe
I think I was. Have we've seen this one in the lower right watch lead?
Yeah, we have.
Okay.
But yeah, let's take a quick snap zoom. I'm sure I'll make.
Excuse me.
Amy happy. And a few others that are working on this group now extensively. So.
Holding.
All right, come on in tight. Let's get this.
Coming in tight. Getting the associate.
Oh, excellent. Good catch.
Is that all you have?
That's full zoom.
Okay. Okay. Come on. Right there. That's what we want.
So, Amy, would you go to Abyssala, Hemicorallium abyssale?
If we get done with this, we might want to tilt camera down just to give you a peek down in that little
and then
patch below us. Let me come out.
Come out a little bit.
that squat lobster,
yeah, I think it's a little.
I believe, is
is that lava?
Munidopsis.
You can come on in.
Thank you.
Yep.
And down.
And so, yeah, that's going to be in the family Actinostolidae. And yeah, Amy just said that she would definitely put that into the genus Hemicorallium. The species is uncertain, and I'm talking about the pink coral now. Excellent, thank you, pilot.
Skid free.
It's lasers.
Yeah.
No, I got them on, Bobby. I was just talking to myself.
Yeah, so we just got the lasers on there, so about a ½ meter in height for that pink coral. And here again we see these piles of what probably is barnacle or some kind of biological material. Yeah, biological, or what used to be.
The upper swing arms forward a little. Are they all the way forward?
I will check.
Are you going to want another ship move
So
here?
there's a few more of these stauropathes.
We have about 13 minutes till we need to start getting ready to come off bottom. We got to be off bottom by 1:14.
That's without any setup time?
Yeah, that's without any setup time, so I figure about 5
So maybe we could take a quick look at that white coral just right of center.
Yep. Coming on in.
Coming in. We got more.
So yeah, that's
Let me just get stable here.
Sure.
another, I believe that's Pleurogorgia. Yeah, we've imaged this one before. Thank you.
All right, come on out, video.
Coming out.
So current depth is 2,345 meters. We have just over 10 minutes remaining on this dive.
Swing arms are fully up.
We'll get you an
Were they
updated
up
position
before?
here in a little
Yes, they
bit.
are. Yes, they were.
Oh, okay. Thank you.
Let's go ahead and do a due north move, Nav.
Copy that. Do you want 30 meters?
Fine.
Okay.
We're gonna have to set up soon anyway.
Bridge, this is Nav.
Let's see what this is. Do a
The
quick
shrimp?
zoom on them. Yeah.
Yeah, sure.
We'd like to request a ship move of 30 meters
Not
due
swimming.
north at 0.2 knots. Good copy.
All right, let's let him go.
And so that shrimp that just drifted across the screen there is likely in the genus Libeus in the family Thoridae.
Fish coming at
Fish.
you.
Okay, come on in, partial. He's coming towards us. Yeah, he was.
Thank you, Bridge.
Okay, come in tight.
And so there's another one of these mercurid, also known as rat tails or grenadiers, Kumba.
Yeah, Dan, it's an interesting observation that this seems to be the most abundant type of rat tail that we see at these depths. And yet that genus is only known from a single specimen collected back in the late 1890s by the vessel albatross. But there's been very little sampling down here, so this thing's so abundant, and yet it's so poorly known.
Yeah, thanks so much for joining, and then for providing that observation. Yeah, so Now, fish are, of course, difficult to sample. They move, and which is why I like to work on things that don't move. But yeah, in terms of what we know about the deep sea, actually, a lot of these places up here in the northwestern Hawaiian Islands were sampled in 1902 by the Albatross, this ship that was by many considered the first large oceanographic research vessel, a vessel that spent time exploring many parts of the world, and it spent about 5 months here in Hawaii, including extended time here in the northwestern Hawaiian Islands. They did close to 350 trawls down to depths of 3,000 meters and collected specimens. And so there's been several things that were collected, over 100 years ago by the Albatross that have not been seen after or just recently seen after. And
sounds like we need to invent or design a fish net or bag.
I'll leave that one to you, Bobby.
I think we could do it. I feel like
quick zoom on this guy.
A quarter of these things fly right into our vehicle, for example.
Yeah. Suction sampler would do it. Maybe. On a fish it'd be tough.
And so I just updated.
Before.
And so that looks like a mysid shrimp. I don't know if Diva is still watching at UHECC.
Alright, I gotta let him go.
So it looked to me like a mysid or a possum shrimp. So they're named a possum shrimp because they have this little brood pouch. And so yeah, just updated our position, and we've traveled about 750 meters. And another point here that's very interesting about Bruce's comment about the Albatross.
Got about 5 minutes, pilots, before we
one of the very valuable things is everything that was collected back then. Everything was properly documented and deposited in museum collections. And in fact, a lot of those specimens are archived, or all of them are archived at the National Museum of Natural History, the Smithsonian Institution, and are still available to
do a quick
be
zoom on this glass
studied to this day. We just described a species of black coral last year from those collections, as have many other scientists and many other groups. And so that's something that we're hoping to do with this project as well, is collect limited amounts of specimens, deposit them in depositories where they can be studied in perpetuity, because there's a great wealth of information that can be learned from them. And so this is another dead sponge.
I was drifting in, trying to. Yep.
It could be either in the family Euplectellidae.
There we go.
Yeah, so it is in the family Euplectellidae, and there's two genera that have this kind of morphology. There's the Regadrella, and Dictyalis. So Dictyalis is characterized, they have these tiny spines that are kind of project outwards from the sponge. Of course, this is now dead, and we don't know how much of
Right, we can come out.
has decayed. So we'll leave it at the family level for now.
Oh, wow.
A nice little community right here, a pink coral, another chrysogorgid coral, several black corals in front of us. Yeah, very nice aggregation of animals.
I just want to remind everybody that is still joining us that this ridge that we're diving on is actually, the surface of this is approximately 25 million years old, and the preservation of these pillow lava flows, the fact that they're still intact as we can see currently in the view is simply amazing. And we've gathered a lot of beautiful landscape images along this entire ridge, and some of the intact lava flows have led us right up onto a local high, which was the volcanic vent in which these eruptions had come from. So an amazing dive, and very successful in discovering these high-density coral and sponge beds, as well as beautiful volcanic scenery along the entire way, and 4 very successful sample collections due to our very talented ROV team.
Yeah, I couldn't agree with you more. Just a spectacular dive. We got all our collections, so we covered the ground that we wanted to cover. Got some beautiful close-up imageries. Yeah, just absolutely beautiful.
Packed with corals now.
And so now this is the first time we've seen this guy, this coral on our right. So that's another pretty conspicuous component. So that's Iridogorgia magnispiralis, or large spirals. Individuals of that species have been imaged that are over 5 meters long. So in fact, individuals of this same species have been measured to be the largest Gorgonian coral known in the world. So some of these individuals have been measured to have the same height as that of an adult giraffe. So that gives you a little bit of an appreciation of, that there's a lot of life down at this depths, and there's a lot of big life. And this is a lot of times we get these questions about why are we spending time here? Why to explore these habitats at deep depths? These are the largest habitat on Earth. Most people probably realize that the vast majority of the planet is covered with water. But what most people don't realize is the vast majority of that ocean surface is in very deep water. So the average depths of the ocean are over 3,000 meters deep. And so it's places that we know nothing about. So fundamental processes about how the planet works and how life works, the answers are likely to be found down here in the deep water, because this is the most common habitat on Earth.
There's no rush to start moving the ship, so until we feel that we're safe.
The other thing is we could see if this is the heading the ship wants to be on for recovery.
So, yeah, this is a Iridogorgia magnispiralis.
If you wanted to, maybe they want to point 90 degrees out.
Hey, how's that, Ed?
Just want to.
Kind of fun, just
we have about a little under 5 minutes remaining here on this dive.
All right.
And just want to once again
Bobby,
thank
all
of our shore-based scientists for participating, providing very valuable help with IDing things, guiding our observations. Truly appreciate that. And I also want to thank all your folks that joined us at both over the live streams as well as at the many general displays.
Doesn't sound like they indicated they want to make a move either way.
Including folks at the Maui Ocean Center in Maui, the Waikiki Aquarium on the island of Oahu, as well as the Mokupapapa Discovery Center on the island of Hawaii.
Thank you, Ed.
Sure, this is an auto iris.
So this is live video footage being broadcast to you from NOAA's premier exploratory research vessel, the Okeanos Explorer, and this is video footage of a remotely operated vehicle diving in places where we haven't dove before, looking in places where we haven't looked before, and making new discoveries. So actually, can we just take there's that sponge right straight ahead.
Sure. Well, Bobby's stowing my swing arm, so.
Okay.
Can I go ahead and just try and get
So
what
yeah,
I can with the
I
light?
can get closer to it.
Fair and
before it leaves.
day. Okay. Yeah, that's all I needed.
That's all you needed?
Yep.
I mean, we have a couple minutes here.
Well, we hadn't imaged that guy on this dive, so.
Okay, let's get it. I think Bobby could still keep stowing.
All right, go ahead. If you have any more, Ed.
Yeah, so that's another one of these glass sponges. The best we've been able to go to these are to family, the fair and I'm at today, and so these are characterized by these kind of hook-shaped attachments that they have to the substrate that's kind of hidden in view now.
All right. You can keep you still stowing. Okay.
Excellent, thank you.
Feels like we're drifting in.
Yeah, we are.
So yeah, that gives you an appreciation for the very porous tissues that sponges have. So that's what gives them their common name of sponges. This kind of spongy tissues. They're some of the most, the simplest animals known.
Yeah.
No real nerve or tissue layers. They do have these specialized cells that are, their only job is to pump water through their tissues.
That is
And that way they can accelerate their feeding. And nice beautiful shot here of several chrysogorgid corals. These piles of this, what we believe is barnacle material wedged between the rocks.
All happy, Bobby?
All happy.
So, if I turn to port and go underneath you, we're going to have to come up a little bit, though.
So we're just about to get ready to
Swing arm stowed, drawer reclosed.
To store all the
We'll
equipment
adjust
and push off the bottom.
shilling once we start ascending.
Our
Hydraulic secure.
last depth here is 2,343 meters.
Lasers are still on.
So,
And I'm putting,
yeah, it's truly been a spectacular dive, and I really want to thank everyone. Pilots did a tremendous job securing 4 samples that are going to be extremely invaluable to several researchers.
On ship's heading.
So we collected all the samples that we wanted to collect. We covered the ground that we wanted to collect, got some beautiful close-up imagery.
No rush.
Got to some of these places with high densities that we wanted to get. So it's really been a wonderful dive. So yeah, thanks to all our shore-based participants. And thank you all for joining us. So it's now 3:15